Good day, everyone. Today we are going to study all about the structure, especially with the bacterial cell structure. So we now move on with your prokaryotic cell structure. So take note that your prokaryotic cell is simpler than the eukaryotic cell at every level. So with only exemption that the cell envelope is more complex. Okay, take note that the cell envelope is more complex. So your prokaryotes have no true nuclei. Instead, they package their DNA in a structure known as your nucleoid. So your nucleoid is associated proteins exist in bacteria and are distinct from the stones in eukaryotic chromatin. So the negatively charged DNA is at least primarily neutralized by small polyamines and of course your magnesium ions. So also it is an ellipsoidal region, okay? Your nucleoid is an ellipsoidal region that contains cells, chromosome, and of course, the numerous proteins are present in your nucleoid. So, your prokaryotic cells are surrounded by complex envelope, yan yung sinabi natin kanina, in which they differ in composition among the major groups. So, these structures or yung cell envelope natin protect the organism from hostile environments such as your osmolarity, harsh chemicals, and even antibiotics. So, this is the reason why when it comes to the bacterial envelope, they are the ones protecting the cell or yung bacteria in terms of our antibiotic treatment. So we also have your plasma membrane, in which this is also known as your cytoplasmic membrane. So take note that your cytoplasmic membrane or your plasma membrane is visible in your electron micrographs. And the this unit membrane rather is composing of your phospholipids and 200 different proteins. Okay, tandaan, it contains your phospholipid and 200 different proteins. So therefore, since there are 200 different proteins, the mass of this membrane, approximately 70% of it is your proteins. Okay, tandaan that 70% of the mass of the membrane is your proteins or are your proteins. And this is considered to be higher than that of your mammalian cell membrane. Okay, so tandaan that your plasma membrane also is the most dynamic structure of your prokaryotic. So as you can see in the figure, this is a typical picture of your plasma membrane in which it composed primarily of your proteins and your phospholipids. So mainly ang function niya would be transport, biosynthesis, and energy transduction. Take note that the phospholipids of your bacterial cell membrane do not contain sterols, but instead, they consist yung saturated and your monosaturated fatty acids natin. Okay, tandaan that your bacterial cell membrane do not contain sterols, rather they consist your saturated and monosaturated fatty acid. So, most of your bacteria or many bacteria contain only yung sterol-like molecules natin, which is your hopanoids. So, yung hopanoids natin, they are your sterol-like molecules and most likely, they are the ones stabilizing the bacterial cytoplasmic membrane. Okay, tandaan that your hopanoid, they are the ones stabilizing the bacterial cytoplasmic membrane. Also, we have here a figure of your phospholipid bilayer. Kaya sa tinawag na bilayer, simply because it has your polar hydrophilic glycerol head attached via an ester band to non-polar hydrophobic fatty acid tails natin. Okay, tandaan, meron siyang layer na polar hydrophilic glycerol head and non-polar hydrophobic fatty acid tail siya. So, kaya siya natawag na amphoteric molecule. And your phospholipid bilayer is arranged such that the polar ends of the molecules from the outermost and the innermost surface of the membrane, while the non-polar ends from the center of the membrane. Okay, tandaan that it has hydrophilic, ibig sabihin, polar siya, head. And also sa tail niya, hydrophobic or non-polar siya, hydrophobic fatty acid yung kanyang buntot. Okay, so that's for your phospholipid bilayer. So aside from the nucleolide or plasma membrane, we also have what we call cytoplasmic membrane for your prokaryotic cell, in which these are the following functions: selective permeability and transport, electron transport, excretion of hydrolytic exoenzymes, contain the enzymes and courier molecules that function in the biosynthesis of the DNA, bear the receptors and other proteins of the chemotactic and other sensory transduction systems. So we will discuss one by one each of its functions. So in terms of the permeability and transport of your cytoplasmic membrane, there are actually three general transport mechanisms in which these are your passive transport, active transport, and group translocation. So sinali na lang din natin si special transport processes. So isa-isahin natin yung So the first thing we need to know is your passive transport. So when it comes to passive transport, this mechanism relies on diffusion. Another thing is doesn't require energy, and it operates only when the solute is at higher concentration outside than inside the cell. So basically, your passive transport consists of the three mechanism. We have your simple, facilitated, and of course your channel protein form. So usually for your simple diffusion, it provides neither speed nor selectivity, in which it accounts for the entry of a very few nutrients lang, including your oxygen, carbon dioxide, and of course yung water itself. While for your facilitated diffusion, also uses no energy. In that sense, the solute never achieves an internal concentration greater than what exists outside the cell. When it comes to your facilitated diffusion, this is considered to be selective. Okay, tandaan that for facilitated diffusion, selective po siya. Unlike the simple diffusion that it can neither speed nor selectivity, hindi po niya inaano. Unlike si facilitated na selective po siya. Well, for the channel proteins from selective channels, they facilitate the passage of specific molecules. Okay, si channel proteins, specific molecules yung fina-facilitate niya. So your facilitated diffusion is common in eukaryotic microorganisms but is rare in your prokaryotes. Okay, tandaan that facilitated diffusion is common in your eukaryotic microorganism but rare for your prokaryotes or your prokaryotic microorganism. For this figure, this is an example of your passive transport in which dun sa A natin, as you can see, there is a net movement of gases, yung oxygen molecule natin, from the area of higher concentration to an area of lower concentration, or yung sa outside natin ng cell is considered to have higher concentration into your lower concentration na inside ng ating cell. So as you can see in the figure P, dun nyo nakikita na pumunta na or pumasok na po yung oxygen molecules natin inside the cell. So that's considered to be passive transport in which there is actually a movement of your oxygen molecule from the area of higher concentration to an area of lower concentration. So examples of gases that actually can cross yung ating membranes by passive diffusion would be your nitrogen, oxygen, and even yung carbon dioxide natin. And aside from that, small polar molecules includes ethanol, your water, and urea can also cross yung ating membranes through passive diffusion. Another type of your transport would be your active. So, kung si passive, there are no utilization of energy, hindi kailangan ng energy. Kay active po, from the word active, kailangan po niya ng energy to facilitate yung transport in terms of ions or molecules inside the cell. So, we have your ion-coupled transport and also your ABC transport na tinatawag natin. So, each will discuss one by one. So, we have here your ion-coupled transport, in which this system actually move a molecule across the cell membrane at the expense of a previously established ion gradient such as your proton or your sodium motive force. So, there are three basic types of your ion-coupled transport, which is your uniport, symport, and your antiport ion-coupled transport. So, usually this is particularly common in your aerobic organisms, which have an easier time generating an ion motive force than that of your anaerobes. So, we have the three types. You have your uniport, symport, and your antiport. Sabi ko nga, we have three types of ion-coupled transport. So, the first one is your uniport. Yung uniporters natin, they catalyze the transport of substrate independent of any coupled ion. So, as you can see, si baby blue, mag-move mamaya siya sa kabila in which in uniform or in direction na pare-parehas po. Kumbaga, follow the leader ang magiging peg niya from the outside to the inside ng ating cell. Okay, so as you can see, pumunta na si baby blue dun sa inside ng cell natin in which yung uniport process is solute passes through the membrane in a unidirectional. Okay? Iisa lang po yung direction na pupuntahan nila. So, kung si uniporter is isa-isa silang dumadaan in a unidirection, si ion-coupled transport na symport naman usually catalyze the simultaneous transport of two substrate in the same direction by a single carrier na tinatawag natin. So, as you can see, we have si baby orange and baby blue in which when it comes to symport process or a co-transport na tinatawag natin, there are two solutes that are transported in the same direction at the same time. So, magsasabay po silang pupunta from the outside ng cell to, sabi ko nga, symporters actually two substrate in the same direction by a single carrier po in which gaya na nakikita nyo sa figure is nakapunta na silang dalawa and sabay po silang pupunta inside the cell. So, yung example ng gradient natin would be yung hydrogen dyan that can permit yung symport of an oppositely charged natin or a neutral molecule for that matter. And of course, when it comes to antiport, from the word anti, in which yung ating dalawang baby dyan, si orange and blue, magpapalitan lang po sila. Kaya tinawag silang exchange division yung antiport process natin, in which one solute is transported in one direction, simultaneous po yung isa din na pupunta or transported in the opposite direction. So, as you can see in the figure, nagpalitan po si baby blue at saka si baby orange in terms of their position. So, therefore, when it comes to anti-port process, again, simultaneously po silang into opposite direction na pumupunta or they are being transported in different direction po. So, usually for your antiport, there are approximately 40% of the substrate transported by your E. coli in which they use this mechanism. So again, 40% of the substrate transported by E. coli utilizes yung mechanism na tinatawag nating antiport. So again, review po, kapag uniport, mag-isa po niya or isa-isa sila in one direction. And kapag symport po, parehas po sila, sabay po silang pumapasok. Kapag anti-port from the word anti is, magpapalitan lang po sila ng direction simultaneously. Another one for your active transport would be your ABC transport. So this mechanism uses ATP. ATP means your adenosine triphosphate, which is source of your energy. So therefore, the mechanism utilizes ATP directly to transport solutes into the cell. So these proteins function by transferring the bound substrate to a membrane-bound protein complex. And of course, once there is a hydrolysis of your ATP, it is then triggered the energy that is used to open the membrane pore and allowing the unidirectional movement of the substrate into the cell. So again, when it comes to ABC transport, hydrolysis of your ATP is triggered in which yan po yung nagkakos ng opening of your membrane pore in which this now can allow the unidirectional movement of substrate into the cell. In addition to through transport, we also have what we call your group translocation or your vectorial metabolism. So this will not utilize energy and it is not an active transport because no concentration gradient is involved. So usually in this process, there will be a membrane carrier protein which is being phosphorylated in the cytoplasm, and from there, the phosphorylated carrier protein will bind to the free sugar, and of course, this will release yung sugar phosphate natin. So this system of sugar transport are called now your phosphotransferase system. So kaya tinawag din na phosphotransferase system ang inyong vectorial metabolism. So yung phosphotransferase systems natin are involved in movement towards these carbon sources and in the regulation of several metabolic pathways for your cell. And of course, take note also that for your group translocation, it allows bacteria to use their energy resources efficiently by coupling transport with metabolism. Lastly, we have your special transport processes, in which iron is an essential nutrient for the growth of almost all the bacteria. So under an aerobic condition, iron is generally in the positive or 3 positive oxidation state, and it is considered to be soluble. So another part of our prokaryotic cell that we need to understand is the cell wall. Of course, it is very important structure in which this is very essential for the maintenance of shape and integrity of the bacterial cell. Also, its main function is to provide a strong rigid structure component that can withstand the osmotic pressures which is being caused by chemicals or high chemical concentration of your inorganic ions into the cell, even the attack of our antimicrobial agents. So most of the bacteria cell walls have in common is that it is a unique structure component called the peptidoglycan. So take note that for your peptidoglycan, it has 3 parts: the backbone, which is composed of your alternating NAG and your NAM, or NAG would be your N-acetylglucosamine and your NAM would be your N-acetylmuramic acid, in which this is being connected by your beta-1 for linkages. Also, we have the set of identical tetrapeptide side chains attached to your NAM and a set of your identical peptide cross bridges. So that's what makes your peptidoglycan layer. So we have here the structure of your NAG and your NAM. So as you can see, this is the difference in their structure, like yung sa naka-color yellow na yan, in which we have your OH and that of your yung mga may metal and your OH na yan. So that part is difference in terms of the structure of your NAM and your NAG. And as you can see, this is the components and the structure of your peptidoglycan, in which the chemical structure of your NAG and NAM, the ring structures of the two molecules are your glucose. So glycan chains are composing now of the alternating units of your NAG and your NAM being joined by covalent band. So yan po yung ating alternating subunits of your NAG and NAM. So as you can see yung nabilogan dyan, those are your NAM and your NAG. Digging deeper the structure of our peptidoglycan, we have your glycan chain. So your glycan chain acts as the backbone to your peptidoglycan and composed alternating residues of your NAG and your NAM. So to each molecule of your NAM, so as you can see in the figure, there is an attachment of your tetrapeptide. So kung makikita nyo, yun yung apat na kulay yellow, red, and orange na yan. Those are your tetrapeptide chains in which nag-a-attach sila sa each NAM. So, dun sa taas na NAM at saka dun sa baba. So, yung tetrapeptide na yan, those are your amino acid. Okay? So, tandaan that those are your amino acid like L-alanine, D-alanine. We also have your D-glutamic acid, and of course, it can either be also your lysine or your diaminopylic acid. Okay, so again, we have the four tetrapeptide or your amino acid rather. We have your L-alanine, D-alanine, D-glutamic acid, or either of your lysine or your diaminopylic acid or your DAP na tinatawag natin. So take note on that we have your glycan chain, which is the backbone to peptidoglycan, and it has your alternating residues of your NAG and NAM. So, sa molecule ng NAM natin, there is an attachment of your tetrapeptide chain, which is your amino acids, L-alanine, D-alanine, your D-glutamic acid, or your lysine, and of course, your diaminopylic acid. We also have special components of your gram-positive cell walls, which is most of your gram-positive cell walls contain considerable amounts of your teichoic and your teichuronic acids. And as a comparison in the figure, in the gram-positive, yung left side natin na figure, yun po, meron tayong teichoic acid. However, dun sa ating negative is wala po tayong teichoic acid. So in here we have the different cell structure of your prokaryotic cell, in which we have there yung kanyang components, including the function and of course yung example bacteria natin. So for capsule, this is being utilized for the evasion of your phagocytosis sa tinatawag natin even adherence and as to the attachment. And when it comes to phagocytosis, this is a cellular process in which this is what we call the ingesting and eliminating particles, which is larger than 0.5 micrometer in diameter. And we have the samples like, for example, Streptococcus mutans. And usually when it comes to the staining, we have your, for capsule, usually we utilize your India ink or your nigrosin. As to the cell wall naman po, as we have discussed earlier, is the component would be your peptidoglycan, in which there is an alternating molecule such as your NAG and NAM. And as you can see po, the function is to maintain yung shape and at the same time to resist yung lysis na nangyayari during sa cell po or during the introduction of any chemicals in that manner or even yung antibiotics natin. So, take note that your Listeria monocytogenes, example natin yan yung may cell wall and it's only gram-positive that has endotoxin. So, tandaan that your Listeria monocytogenes is the only gram-positive that has your endotoxin. So, when it comes to endotoxin, that is what we call lipoprotein, lipid A, in which this makes the bacteria more heat stable than that of your exotoxin na tinatawag natin. Its mechanism would be the release of your TNF. Okay, and of course we also have your fibrae, which is a hair-like structure in which this is being utilized for adherence, conjugation, and of course as the virulence factor na tinatawag natin. So when it comes to virulence factor, this is a pathogens or microorganisms ability to cause damage to the host or yung sa atin kapag once na contract natin yung bacteria. And of course, this is defined as the degree to which a pathogenic organism can cause disease. Like for example, yung Neisseria gonorrhea, the virulence factor of your pilus or yung fibri natin. And also, we have your flagellum, which has your flagellin as the protein component niya. And usually its function is to for the motility or yung locomotion na tinatawag natin and tumbling movement. And of course, we have the example like your Listeria monocytogenes in which it has a tumbling motility. And for your spore naman po, we have the component dipicolinic acid which makes our bacteria heat resistant. So, kahit ano pong gawin natin na pagsunog or pag-init dun sa ating mga kinakain, kung may bakteriyang heat resistance po siya, hindi po mamatay. There is what we call survival from heat and chemical resistance when it comes to bacteria that has spore. Okay, so this is a summary of your prokaryotic cell structure.